So, the mind is an embarrassment to science because it is something subjective. And of course, subjective phenomena, we try as much as possible in science to exclude them. Dress it up however you like. You can only ever observe your own mind. It is truly subjective. What the behaviorists did in the previous century was to exclude subjective phenomena from psychology, from the science of the mind. And they focused only on what is externally, objectively observable, namely, of course, behavior. A different approach was taken by this chap, Sigmund Freud. His approach was that science must, the methods of science must be adjusted to its objects, not the other way around. You can't say we don't want to study subjective phenomena. If subjective phenomena exist, if mental experience is part of nature, then we have to find some way of including them within science. We can't say we're not going to study those things. And so what he did was to develop a method of interrogating mental experience, which necessarily meant that it was a highly questionable method because he was investigating subjective things using, therefore, inherently, necessarily subjective methods. And he started his first really systematic study was a study of dreams. And dreams are perhaps, quintessentially subjective because you can't say, I'm busy having this dream, can you see what I see? I mean, I'm experiencing such and such, are you too? And so on. Dreams happen while you're asleep. And you can't report on them. You can only report on them when you wake up. So they're retrospective, single witness descriptions of something that happened while you're asleep. So it really has, all three strokes against it. What Freud did, using the kind of method that I was mentioning earlier, was to get his research participants, who were mainly his patients, to take each element of the dream as they remembered it and say everything that came into their heads in connection with that image. This was the famous or infamous free association method. And Freud said, if you do that, what you find is that the associations aren't random. They all sort of point to a underlying meaning that although the dream on the surface seems chaotic and nonsensical, the associations lead you to a latent meaning, what we would nowadays call an implicit content. Freud called it latent content. So the explicit dream, what Freud called the manifest dream, is chaotic and random and apparently meaningless. But Freud said, if you follow this method of his, you find a latent, implicit content. And this content is of a stereotyped kind. It is in the nature of a wish, a heartfelt wish, a wish so heartfelt, revealing desires and inclinations of a kind that you normally wouldn't want to admit to during waking life. And on the basis of this questionable method and surprising claim, Freud built the whole of psychoanalysis. In his book on dreams, which was published in 1900, he made this little diagram, and this became the basis for his whole theory of the mind. Freud said that normally we perceive things during waking life, we lay them down in memory, and eventually in unconscious or implicit memories. And then we draw upon these to have our rational plans, goals, strategies, thoughts, which we then act on. This was his sort of picture of the mind. And on the basis of what he claims to have observed using his free association method in studying dreams, he said, well, if this is how dreams work, then the mind must be structured something like this, something like what I have on the screen. And then he said, because it appears during sleep, this part of the mind, what he called the preconscious, the rational verbally encoded thought processes which lead to our goal-directed actions, this goes to sleep, more or less. It doesn't go to sleep completely, as any parent of a little baby knows. There's a little kind of antenna that stays awake, but for the most part, this rational thinking, goal-directed part of the mind goes to sleep. And Freud said what that does is it releases from inhibition these heartfelt wishes and desires that I mentioned earlier, which are normally unconscious. That's what this little UCS stands for. And so these unconscious, disreputable desires press for action, press for discharge in motor activity. And so for that reason, says Freud, this is blocked. Motor activity is blocked during sleep, so that we cannot act on these, what he called repressed desires. Because why? That would wake us up. This is a very important point. He says it would wake us up if we acted on these desires. So instead, the mental activity that occurs during sleep, the activation or the release from restraint of these unconscious wishes and wants, they progress in a regressive direction, in a retrogressive direction, back onto the perceptual systems. And so we have an hallucination. We imagine ourselves doing things rather than actually doing things. So the function of dreams, says Freud, is to protect sleep. In order to not have to actually act on these desires, we hallucinate ourselves in the virtual reality of the dream to be fulfilling them. I'm sure I'm not alone in wondering, well, why don't my dreams work like that? I don't seem to be fulfilling all of my desires, my heartfelt secret wishes during my dreams. And Freud had an answer for this. He said, well, that's because, as I told you a minute ago, this part of the mind, the preconscious part, isn't entirely asleep. There's still a glimmer of a watch person there checking on what's going on. In the outside world, it might be a baby crying. In terms of the inside world, it is, oh my God, what is this thing that's happening? I seem to want to do that. Oh my word, that too might wake the sleeper up. And so it's disguised, says Freud. There's a censor that disguises and distorts these wishes, and this is why dreams look so nonsensical. They're not transparent. They're censored. Now, that's a very complicated theory. And remember, the method upon which it's based is simply having the dreamer say everything that comes into their head. On the basis of that method, Freud made these really quite bold claims. Despite the sort of shakiness of the method and therefore the shakiness of the theoretical conclusions, many people were persuaded. And the psychoanalytic movement became very powerful in the early years of the 20th century, so much so that it came to dominate world psychiatry. And in America, which those days dominated the world, psychoanalysis was absolutely de rigueur. You could not become a tenured professor at any decent American university, a professor of psychiatry, unless you were a psychoanalyst. So against this backdrop, the publication of a book called Conjecture and Refutation by the famous philosopher of science, Karl Popper, was rather a blow to psychoanalysis and to psychiatry, because Popper used this theory that I've just described to you as an example of what he called pseudoscience. It looks like science, it sounds like science, it aspires to be science, but it isn't science. And the basic point that Popper made was it does not give rise to predictions which are falsifiable. This was Popper's basic rule of how to determine whether a theory is scientific or not. If it's scientific, it must give rise to predictions which can either be confirmed or disconfirmed. And Popper said in the case of Freud's theory, you can't disconfirm it, because when you say, but this dream and this dream and this dream aren't wishful, I don't see any heartfelt desires in these, Freud would say, well, yeah, that's in the manifest dream, the explicit content, but behind that is an implicit latent content, which I interpret. And Popper made the not unreasonable point that you can always interpret every dream to reveal some hidden wish that you find as somebody might find meaning in tea leaves or in an ink blot. But that doesn't mean that it's really there. And the crucial point is you can't falsify the claim, because every dream can be made to support the theory, the hypothesis that dreams are an attempt to fulfill a heartfelt but repressed wish, and that the purpose of all of this is to protect sleep. So bearing in mind how influential psychoanalysis was in world psychiatry at that time, it was a great relief when in the early 1950s, two American physiologists named Azarinsky and Kleitman discovered what has come to be known as REM sleep, rapid eye movement sleep. They used a new method, a method that wasn't available in Freud's day, called the electroencephalograph, EEG. And this is a technology which you place electrodes on the scalp of the research subject, which records through the cranium the electrical activity going on in the brain, the level of electrical activity, of brain arousal. And as Azarinsky and Kleitman predicted, that if you measure the brain by this method as it goes to sleep, its level of arousal should decline as measured by the EEG. So what you see on this sort of cartoon here, this axis is the level of brain arousal from wakefulness going to lower and lower levels of brain activation. And this axis here is time. So we're looking at 8 hours of time here. And what Azarinsky and Kleitman observed was exactly what they predicted, that as you fall asleep, so your level of brain arousal declines. What was surprising, though, the wonderful thing about science is that you don't only find your Easter eggs where you had hidden them beforehand, you get surprises. The surprise was that 90 minutes later, the brain reactivates itself and then goes down back into that quiescent state and then reactivates itself, and does this in regular, roughly 90-minute cycles. So what was this all about? You know, it's a paradox, and they originally called it paradoxical sleep before it was renamed REM sleep. The paradox being, you're asleep in a state of repose and quiescence, and yet your brain becomes almost as activated as it is when you're awake. This led them to the hypothesis that perhaps this paradoxical arousal of the brain was the physiological equivalent of the psychological paradox that during the unconsciousness of sleep, you have these moments of consciousness where, despite the fact that you're asleep, you suddenly start experiencing something, and that is the dream. This paradoxical punctuation of the unconsciousness of sleep with the conscious experience of dreams, they hypothesized might be the subjective correlate of the objective phenomenon they just discovered, namely this paradoxical brain arousal. So that led to a further experiment done by not Azarinsky and Kleitman, but the unfortunately named Dement and Kleitman. They predicted that if you wake people up during REM sleep and ask them if they're dreaming, as opposed to wake them up during non-REM sleep, during REM sleep, you will get dream reports. and during non-REM sleep, you will not. And that's exactly what they found. In their mid-1950s paper, they reported that approximately 90% of awakenings from REM sleep produce dream reports, and less than 10% of awakenings from non-REM sleep. And that's about as close to a perfect correlation as you could hope for, given the nature of the phenomenon we're talking about. Subjective first-person reports from waking people up, in the middle of the night, and dreams are, of course, infamously forgettable. So that was really a striking confirmation of the prediction that flowed from their hypothesis that REM sleep was the objective equivalent of the subjective state of dreaming. Now, this was great because now we had something objective. Remember, psychiatry was dominated by psychoanalysis. Psychoanalysis was based on this free association method and fundamentally on Freud's findings about dreams. So at last, we had the possibility to be able to get out of that corner. And now that we had this objective physiological phenomenon of REM sleep, we could get a proper handle on what really drives dreams. This was made much easier by virtue of the fact that it's not only human beings who have REM sleep, all mammals do. And why that was important is because the standard way of going about determining which part of the brain is generating a particular function is to lesion, that is to say, to experimentally damage the different parts of the brain and see which part, when damaged, leads to a loss of the function at issue. So a chap named Michel Jouve led the charge using cats. What I have on the screen now is a human brain, but human brain is sort of shockingly similar in its anatomy to a cat brain. And perhaps why Jouve focused on cats was because they sleep so much. And what he did was really quite horrible. He sliced through the cat brain, progressively making deeper and deeper cuts to see at what point do the poor cats stop displaying REM sleep. And the surprise in this instance was eventually Jouve made a cut here, separating the brain stem from the whole of the forebrain. The forebrain, including the cortex, is the organ of the mind. And remember what Freud was saying, that dreams are driven by these deep-seated desires and wishes and wants and so on. These are clearly mental forces. But when Jouve made the cut here, effectively disconnecting the organ of the mind from all that remained, the brain stem, which is a very primitive structure, which was considered to be entirely mindless, sort of responsible for orchestration of bodily functions and reflexes. REM sleep continued in these cats, even though the forebrain as a whole had been disconnected. And it was only when Jouve cut right down here to the structure which is known as the pons, it was only when he reached that level that REM sleep stopped in these cats. And so that showed that whatever it is that drives dreams, it's occurring at this level. Jouve then passed the baton on to his pupil, Alan Hobson, and Hobson was able to identify precisely what it is that's going on in the pons. When we go into REM sleep, there are a group of cells over here in a region called the mesopontine tegmentum. These cells release a neurotransmitter called acetylcholine, which activates the forebrain. And then when we go out of REM sleep, these cells stop firing, and another group of cells, two other nuclei here, which I don't need to go into the details of, they start firing. So when these ones are on, those ones are off. When these ones are on, those ones are off. So what is it that causes REM sleep, dreaming sleep? It's a simple switch, on, off, and it switches on and off every 90 minutes, automatically. In the mindless, reflexive part of the brain that we share with all vertebrates. Fishes have the same structures down here as we do. And so Hobson wasted no time. We've now reached the 70s. Jouve's findings were the mid-60s, Hobson's were the mid-70s. He wasted no time in pointing out that Freudian dream theory was clearly wrong. This part, the acetylcholine, there's nothing motivational about it. There's nothing emotional about it. There's nothing mental about it. It's just randomly firing neurons in the cortex. The cortex, being the organ of the mind, is now activated. And so you have this chaotic, meaningless, mindless mental activity, conscious experiences, thoughts, memories, and whatnot. Hobson said that all that the cortex can do is join these dots, sort of make some kind of episode out of this random firing. To quote him, he said, "It makes the best of a bad job." And that's the dream. So this was called the activation synthesis theory of dreams. These pontine cholinergic, the cells that release acetylcholine, they activate the forebrain, and the forebrain, poor thing, synthesizes all of these inherently meaningless images, experiences into a dream. I say inherently meaningless. It's really important to get this point, that the thing that causes the dream, says Hobson, is this automatic pre-programmed clockwork of the firing of these cholinergic cells, which randomly activates the cortex, and the cortex joins the dots. You can come and look at that just as with an ink blot after the fact. You can look at this random activity, just like with tea leaves. You can say, makes me think of this or that. It suggests the following to me, but that meaning is not inherent in the tea leaves. It's not really there. It's not in the ink blot. You project it onto this otherwise actually intrinsically meaningless, mindless data. So that was Hobson's theory, the activation synthesis theory, and he presented it at a meeting of the American Psychiatric Association in 1977. After his lecture, a vote was taken among the members, and the question that they were asked to vote upon was, in light of Alan Hobson's findings, is Freudian dream theory still scientifically tenable? And not surprisingly, the vote went against Freud in Hobson's favor to the tune of 2-1. The ratio was 2-1 in Hobson's favor. And this was a decisive moment in the demise of psychoanalysis as a credible scientific foundation for psychiatry. 10 years after that, well, a little bit less than 10 years after that, I started the research program that I'm now going to report to you. What I did, I am a neuropsychologist rather than a neurophysiologist. And so I was more interested in the cortex than the brainstem. And this was in the mid-1980s. And we knew that the cortex does something rather more complicated than synthesize. This idea that the cortex is activated randomly during Rem sleep, and what it does is join the dots. That's reasonable enough, but the different parts of the cortex contain different kinds of dots and do slightly more finessed operations than synthesize. So I predicted that, for example, if you have damage in the visual cortex, then the visual content of your dreams will be distorted or perhaps obliterated. That if you have damage in language cortex, that you won't be able to speak in your dreams, just as you can't speak in waking life, and so on. These are sort of fairly obvious predictions. So I wasn't questioning Hobson's theory. We all knew that the problem of where and how dreams are generated, the causal mechanism of dreams was in the brainstem. It was the same thing as the mechanism of REM sleep. But I was interested in the contents in what does the cortex actually do that unpacks this notion of synthesis. What I found was really mostly, well, not mostly, it was entirely, in fact, serendipitous. The only one of my predictions that I think is theoretically interesting that was confirmed, I predicted that the prefrontal cortex, which I'm indicating on the screen now, this area here, which is our evolutionary pride and joy as human beings, we got lots of prefrontal cortex. It's responsible for, it's equivalent to this pre-conscious part of the mind that Freud spoke about, the sort of verbally mediated, logical, rational, goal-directed agency, the sort of head office of the mind. I hypothesized, since during dreams, we don't seem to be in charge of our mental processes. They don't seem particularly rational and goal-directed. We are kind of passive recipients of what happens to us during dreams. I predicted that damage to this part of the brain would have no effect on dreams. And I was right about that. If you take the dreams, these patients with severe damage to the prefrontal cortex, they are very impaired in their waking cognition, but their dreams are indistinguishable from yours and mine. Blind raters cannot find any difference. They cannot differentiate the dreams of patients with damage to the prefrontal cortex and the dreams of you and me, assuming that you, like me, do not have prefrontal cortical damage. Here's a sort of summary of what I found. What I've just told you about was this part here, this dark blue area on this diagram is the prefrontal cortex here. As I said, damage there has no effect on the content of dreams. Now I'll tell you about some, I'm not going to tell you about everything I found, we don't have time. I'll tell you about the two most interesting things. And these were, as I said a moment ago, entirely unexpected chance discoveries, just good fortune. Patients with damage here, which is the mesocortical tegmentum, in other words, patients with damage here, although they lost REM sleep, and in fact, we knew before my study that human beings with damage there, like cats and rats, REM sleep is obliterated with damage there. I was not studying rats and cats, I was studying human beings, so I could talk to them. And of course, I didn't produce the damage in my research participants. I had to wait for naturally occurring lesions. I studied a large group, roughly 350 patients, so it took me several years to collect this data. I had 18 patients here, all of whom, like previous research had shown, these patients do not have REM sleep, and yet my patients reported dreams because I was using subjective methods, in other words, talking to people about their experience. I was absolutely shocked to find all these years after the discovery of REM sleep and its correlation with dreaming in the mid-1950s, this correlation was, taken. The fact that REM sleep and dreams occur together at the same time, this correlation was taken to be an identity. It was taken to mean that dreams and REM sleep are the same thing. As I said to you earlier, REM sleep was taken to be the objective manifestation of the subjective process called dreams. And we then dropped the dreams like slippery fish because they're an embarrassment to science. You don't want to study this fluffy subjective stuff. So we then shifted to just studying the objective brain processes in order to get a proper scientific grip on what are the causal mechanisms of dreams. And so to my amazement, when I discovered that my 18 patients that I actually spoke to and gathered subjective reports from, reported, all of them, that they were still dreaming, I went back to the literature over the last 40 years and found that nobody had ever done that. Nobody had ever, although we knew that human beings, like other mammals, when they have damage here, their sleep stops. Nobody had bothered to ask them, are you dreaming? And I really think it's for the reason that I've been saying periodically during my talk so far, it's because subjective data, we have an aversion to it in science. But it leads us to wrong-headed conclusions to leave out the subjective data, because the fact that dreams and REM sleep correlate does not mean that they're identical. You know, we teach undergraduate students this. Correlation is not causation and all that. So by leaving subjective data out, we can make some dreadful mistakes. It seemed as if REM sleep and dreaming were not the same thing, because damage here leads to a loss of REM sleep with preservation of dreaming. Strengthening this conclusion was the fact that patients with damage to posterior cortex, patients with damage here, these patients had the opposite phenomenology. They had continuation of preservation of REM sleep, but loss of dreams. So with damage in the brainstem, you have loss of REM sleep with preservation of dreams. With damage in this cortical region here, you have loss of dreams with preservation of REM sleep. This is called a double dissociation of function. It showed that they cannot be the same thing. Dreams and REM sleep are two different things. Now, this part of the brain is responsible for, in fact, if any part of the cortex performs the function that Hobson called synthesis, then it's this area, because this is called the inferior parietal lobule. It is where somatosensory information and auditory information and visual information converge. So if you're going to generate a multimodal mental image or hallucination, such as we do in dreams, then no region would be more likely to perform that synthetic function, that bringing together of all of our different perceptual modalities into an experience than this part of the brain. So that wasn't a surprise. What was a surprise, and this is the last of my findings I'm going to mention to you, was that damage in this area had the same consequence. The area that I've marked red. This is the white matter, not the cortex, the white matter, the fibers under the cortex in this part of the brain here, which is called the meso, sort of like the ventromesial quadrant of the frontal lobes, the bottom middle part of the frontal lobes. And unlike this area here, which made good sense that damage here, if you can't generate a mental image, how are you going to generate a dream? The same thing could not be said of this region. It was not by any means clear why damage there should lead to a loss of dreaming. I only had 9 patients, 9 out of my 300, and I think it was 361 cases I ended up with, and only 9 of them had. It's quite hard to get bilateral damage here. So it wasn't surprising that I had such a small number of them, but you can't, 9 is not enough. So the area of damage here are these are facsimiles of the scans in those very 9 patients that I've just mentioned. When I looked at these scans, I was reminded that this area is where we used to do an operation called prefrontal leucotomy, which was, with gay abandon, this operation was done in the 30s and 40s of last century for the treatment of psychosis. That is to say, the positive symptoms, hallucinations and delusions were well controlled by this operation. I went back to that literature because in the 1930s and 40s, when psychoanalysis was all the rage, psychiatrists used to talk to their patients about their dreams. And so I thought, well, those psychosurgeons presumably will have observed the same thing that I did. In my measly 9 patients, they performed this operation on thousands of patients. So I went back to that old literature, and to my relief, I found that what they reported was the operation had 3 effects. One was the patients no longer had hallucinations and delusions. The second was that they lost spontaneity and motivational drive. And the third was that they stopped dreaming. In fact, it was so widely reported that, so generally recognized that these patients lost dreams, that one of the scientists concerned, his name was Schindler, observed that if the patient after the operation continues to dream, this is a bad prognostic sign. It means that the operation hasn't worked. And that made a kind of intuitive sense to me, because if you can generate a dream, that means you can generate an hallucination. In fact, dreams are not only hallucinatory, they also are delusional. You believe things which are not true during dreams. And so if you can generate a dream, then presumably you can generate a psychotic symptom. And this led me to the path of answering the question, which was necessarily arose from the findings I've just reported to you, which was, what is it that's going on in this part of the brain that's so fundamental to the process of dreaming? Clearly, damage here, as not only I observed in my little handful of cases, but as had been observed in so many cases in the 1930s and 40s, the damage here leads to a loss of dreaming. But this is spaghetti junction, this part of the brain. There are millions and millions of fiber pathways going through this region. Which one was responsible for generating the dreams? And I hypothesized that it's this one. This is called the mesocortical mesolimbic dopamine circuit. And the reason why I hypothesized that it was this one is because that operation, the prefrontal leucotomy, it was dropped in the 1950s, not for ethical reasons, but because we had discovered a drug. We had discovered antipsychotics, and antipsychotics block dopamine in this pathway. So since this pathway courses through the very region where that cut was made, I thought this was most likely the critical pathway for generating dreams, as it is for generating psychotic symptoms. So from this hypothesis flows predictions. Those predictions are testable, a la Karl Popper. And so, I mean, I knew that cutting through this pathway led to a loss of dreams, but there are many other pathways there too. So it led to the prediction that if you chemically manipulate this pathway, if you increase the level of dopamine activation in this pathway, this should increase the number and frequency, etc., of dreams, and it does. A study done by my colleague Ernest Hartmann showed that if you give patients levodopa, a dopamine booster, they have longer dreams, more frequent dreams, more emotionally intense dreams, more bizarre dreams, more nightmare. Every single dream measure is increased, but not REM sleep. It's something separate. It's the dream that's increased. And my student Kelvin Kai-Ching Yu showed that blocking of dopamine by antipsychotics leads to the opposite. Less frequent dreams, shorter dreams, less emotionally intense dreams, etc. So that was one method whereby we tested predictions arising from the hypothesis that this is the circuit that generates dreams. There was another study using a different method. This is positron emission tomography. And this was a study done by my colleague Alan Brown. And he showed, in fact, it was remarkable, the coincidence between his imaging findings and my lesion findings. You will see here the prefrontal cortex, which, as I said, is switched off during sleep, evidently because of the quality of dreams, and my lesion findings showed that damage here has no effect on dreams. Well, his imaging studies showed that this part of the brain is not active during dreaming sleep. Dreaming sleeping, REM sleep, remember 90% of awakenings from REM sleep, dreams do correlate with REM sleep. So you can operationalize dreaming sleep as REM sleep. And this part of the brain is not activated. But much more interesting, oh, and also this part, the imaging that I said, the synthesis in the parietal cortex, this part is activated. There you have it. But much more interesting is this. This is clearly the main event during dreaming sleep. And this is where that mesocortical, mesolimbic dopamine pathway is located. It is the most active part of the brain during dreaming sleep. So Lesions suggest that this is the part that's responsible. Pharmacological studies suggest this is the part that's responsible. Imaging studies reveal the same. And so did these two studies by Dahan and colleagues and Lehner and colleagues. What Dahan showed was that he measured with single cell electrodes, he measured the activity in these neurons, the source cells of this circuit, and they fire in in bursts like that, which is the most active that a neuron can be, is when it shows burst firing. So he found prominent burst firing of these neurons during dreaming sleep. And Lehner's study measured by a method called microdialysis the release of dopamine at the terminals of this circuit. And the release of dopamine is maximal during dreaming sleep. So all of these different methods, the predictions arising from them, from my hypothesis, were confirmed by all of these methods. And so it seemed like a fairly reasonable, solid conclusion that this was indeed the circuit that drives dreams. I must just mention something about Hobson in passing. Hobson, as you know, had found that cholinergic cells here fire during REM sleep. So on his hypothesis, if you test it in the way I showed you, that a lesion here does not lead to loss of dreams, but a lesion here does. The second of the methods that I mentioned, the pharmacological probes, if you block dopamine, it blocks dreams. What happens if you block acetylcholine? The interesting thing is the very opposite of what Hobson's theory would predict. You block you give anticholinergic drugs, patients have more dreams, not less. In fact, they have dreams while they're awake if you're not careful. So all the evidence pointed to this being the circuit that drives the dream process. So Hobson invited me over to his lab in Harvard to present my findings, and I did so. And he was very, reasonable, decent, accepted the evidence. And at the end of my presentation to his team, I said, Alan, I think you owe Freud an apology, because, if any neuroscientist was to look at this brain and say, how would you expect, if Freud's dream theory was correct, what would you expect to see? Well, Freud says the rational, sort of verbally mediated thinking brain goes to sleep. Well, that's what we see. And it releases from its normal constraints this emotional driven, wishful part of the brain, which is what there's no part of the brain that's more wishful than the mesocortical mesolimbic dopamine system, as I'll show you in a moment. And there's regression onto the perceptual systems, just as Freud hypothesized. So the dreaming brain looks pretty much like what you would think on the basis of Freudian theory. And the evidence that you adduced against Freud, namely that REM sleep works entirely automatic clockwork, switch on off every 90 minutes in a mindless part of the brain, that drives REM sleep, but it doesn't drive dreams. When I went back to London, which is where I was living those days, Hobson sent me an email. In fact, it was the early days of email. And he said, Mark, thanks so much for your presentation. As you know, I find your evidence very compelling, and I'm willing to acknowledge what you found. But did I hear you correctly at the end? You seem to be saying that these findings support Freud. Because if you're going to interpret your findings as being supportive of Freud, then I'm afraid I'm not willing to acknowledge your findings. So much for objectivity in science. I was absolutely shocked. So, I was then invited, he and I were invited to present our respective findings at the biannual Consciousness Congress, which was held in Tucson, Arizona. I can't remember the name of the organization, something like Scientific Study of Consciousness or something. They have this famous meeting every 2 years. And we were invited to debate, to present our respective findings. And then the audience was asked to vote on this very same question that was raised in the 70s in the American Psychiatric Association. In light of these findings, is Freudian dream theory viable or not? And on this occasion, 2006, almost exactly 30 years later, our assembled colleagues on this occasion voted 2-1 in Freud's favor. 30 years later saying, well, these findings, they are compatible with Freudian theory. Now, Hobson, well, let's leave Hobson out of the rest of the story. This is really surprising. I mean, remember the method that Freud used, just talking to people, just getting them to say whatever comes into their heads. And he discerned from this, that something like this goes on, that this activation, that this goes to sleep, that leads to release from inhibition of this, that threatens sleep, so you might wake up. And so Freud says, so it regresses onto the perceptual systems. I mean, this really did look pretty much like what's going on. But it looking pretty much like hardly tests Freudian theory. It's just saying the reasons why we rejected the theory had themselves been rejected. That doesn't mean that the theory is correct. It was remarkable, the sort of the synergy between what we were finding about the dreaming brain and what Freud had inferred from these conversations with his patients. But that's not science. What you have to do in order to do proper science is you have to have testable predictions. And remember, all these methods, I mentioned EEG, but then after that came all these other methods, these pharmacological probes, single cell recordings, microdialysis, positron emission tomography, all of these methods which are available to us today. So we now do have methods which were not available all those years ago, whereby we can test hypotheses, test predictions arising from hypotheses like Freud's. So this is the study that then suggested itself to me. And I've been zooming through the years. I'm now bringing you right up to the present day. So far, we've only done a pilot study on this of the kind that I'm now going to describe to you. And the main study is underway as we speak. At the Charité Hospital in Berlin, we're doing the main study that the pilot data of which I'm now going to report to you. I just want to show you this slide. Oh, wait, I'm going the wrong direction. What am I doing? Oh my word, what have I done there? Wait, here it comes. Here is Panksepp, an expert on that circuit, that mesocortical mesolimbic dopamine circuit. He says if you stimulate this pathway, it leads to the most energized exploratory and search behaviors that an animal is capable of exhibiting. Dahan, who did that single cell recording, he said, what you find in those dopamine neurons, that what happens during dreaming sleep, it's as if the animal is getting its favorite rewards. So the this is this circuit that we're talking about. So I thought, well, so if Freud is right, that released from inhibition, this circuit now lights up like a Christmas tree, leading to the most energized exploratory behaviors that we are capable of, leading to the most rewarding, it's like a consummation of palatable food or whatever it was that Dahan said. Freud said that this threatens our sleep. And that's why we have the regression onto the perceptual systems and we have this hallucinatory experience. So that suggested the following experiment to me. I said, if we take these patients who can't dream because they can't generate the mental imagery, but who still have this drive, this motivational surge during sleep, because they can't have the dream that Freud says protects them against waking up, they should wake up. So there you have a prediction that's testable. Patients with damage here who can't dream should wake up from sleep. They should have more disturbed sleep than patients with damage here who can dream. So that's what I did my pilot study on. And I just make clear, it's a very small pilot study. 5 patients with damage here due to thrombotic infarctions. So they have the same pathology as the patients who do dream. Patients with thrombotic infarctions in this artery who do still dream. I compared 10 of them with 5 who do not dream anymore. And my prediction was in the pilot study that the ones who do not dream, counterintuitively, only on Freudian theory would you expect somebody who does not dream will have worse sleep. You know, the commonsensical view will be, well, you'll sleep better. Your sleep is not disturbed by dreams. But Freud says, no, it's the opposite. The dream is there to keep you asleep. So my prediction was that the non-dreamers would have worse sleep than the dreamers. There are various ways that we measure quality of sleep, which I'll show you in a moment. But before I show you the starter, remember the important thing is that it is falsifiable. This prediction is falsifiable. The prediction that the non-dreamers will sleep worse than the dreamers. If I didn't find that, that would mean Freudian dream theory is wrong. That's the important thing. It renders the theory falsifiable. And that's why I did this study. Now I'll show you my findings. This is a hypnogram. Remember I showed you earlier this cartoon of a hypnogram. This is brain arousal on this axis, and this is 8 hours of sleep in time on that axis. Now these are real hypnograms that I'm showing you here. And this was my first non-dreaming patient. Remember, these are patients with strokes in the perceptual part of the brain. And this is the first one that I put into the sleep laboratory who no longer dreamt. Maybe in the discussion time, if anybody wants to know the methodological details of how do we determine whether somebody really isn't dreaming or not, I can go into that then. So what you see is an hour passes, 2 hours pass, she doesn't fall asleep. Eventually she does, she immediately wakes up again. And then she goes into deep sleep for about an hour. And then for the rest of the night, just can't stay asleep. She just keeps on waking up, keeps on waking up. Whenever she goes into REM, she wakes up, goes into REM, she wakes up. This is called sleep maintenance insomnia, can't stay asleep. This is called sleep onset insomnia, can't fall asleep. It's pretty bad sleep. Here's the second patient. This patient didn't have so much difficulty falling asleep, but staying asleep was really, really hard. And also, whenever the patient went into REM, they woke up. So you see sleep maintenance insomnia, REM, waking up, sleep maintenance insomnia, very bad. I'll tell you what these other things mean a little bit later. I know I only have a few minutes left, don't worry, I'm nearly at the end. This is the worst hypnogram I've ever seen. Many people say, oh, I didn't sleep a wink last night. You know, I'm such an insomniac. I don't sleep at all. You put them in a sleep laboratory, they do physiologically sleep a hell of a lot more than they think they do. But this patient just did not sleep. For 4 hours, they were wide awake, then fell asleep for a couple of minutes and then woke up, fell asleep for a minute and then woke up, fell asleep for half a minute and then woke up, and another half a minute and then woke up and then stayed awake. And then again, just dipped into sleep for fractions of the night. This is just, I don't know what to call it. It's a really terrible, terrible insomnia. I've never seen anything like it. And here's the fourth patient, same thing, New York City skyline, okay, sleep maintenance insomnia. So I'm just giving you this visual impression of what we observed in these non-dreaming patients, exactly what was predicted, terrible sleep. Now I'll give you a quick summary of the dreamers, 10 of them, compared to the non-dreamers, 5 of them. Remember, this is just the pilot study. How much time did they spend asleep? Remember, these are all stroke victims. They're very ill people, so you don't expect them to have brilliant sleep. They've recently suffered a stroke. But the dreamers, on average, had 375 minutes of sleep. The non-dreamers, 267 minutes. That's a big difference. So the dreamers spend 3/4 of their of those 8 hours of sleep. The non-dreamers spend about half of those 8 hours of sleep. So it's about a quarter, about 25% difference, which is a big difference. this is a standard way in which we measure sleep efficiency. I won't go into all of this, but you see, again, a 20% difference in sleep efficiency using the standard measures between the dreamers and the non-dreamers. This is an awakening index, which is the number of times that the patient wakes up per hour of sleep. Because remember, they have very different amounts of sleep. So per hour of sleep, how many times do you wake up? The non-dreamers wake up 3.5. Remember, these are ill patients. They wake up 3.5 times an hour. The non-dreamers, 12.5 times an hour. Massive difference. This is arousal. These are brief awakenings, not waking up and staying awake, but waking up briefly. Again, look at the difference. 48.5 arousals per hour of sleep in the non-dreamers, 19 arousals per hour of sleep in the dreamers. Massive difference, 30 percentage points. Micro arousals are very brief. You actually, they're little like micro awakenings, less than a minute. Those were the things I was showing you on the hypnogram. I said, I'll explain to you later what they are. Again, look at the difference. It's absolutely massive. This is why I'm showing you pilot data, because it's clear that here we are seeing something biological. This is not some sort of statistical blip. This is a massive difference. So again, the microarousals, the dreamers have 15.5 microarousals per hour of sleep. The non-dreamers, 39 arousals per hour, microarousals per hour of sleep. So every measure that we used, every prediction that arose from the hypothesis that dreams protect sleep was confirmed in this little study. And as I say, we're still busy completing the bigger study. Whatever we find in the bigger study, the crucial thing is, please note that the theory is testable. So, we include dreams, we include subjective phenomena in science. It's a hell of a lot easier to do it now in neuroscience when we can study simultaneously the objective physiological state of the brain and get subjective reports. If everybody reports the same thing subjectively when the brain is in the same state objectively, well, there's every reason to believe the subjective reports. So we can bring subjectivity, we can bring the psyche back into neuropsychology today. And so what we have found for the first time, although I must emphasize I'm giving you preliminary data, it seems for the first time we have found a biological function for dreaming. It's been a cause for great consternation. We spend 20% of our sleeping hours dreaming, but it's never been apparent, it's never been possible to determine why. What is all of this dreaming for? So whatever else it's for, one thing that we seem to be finding is that dreams protect sleep. If you've had any difficulty following my argumentation, you should be ashamed of yourself, because look at this last slide. This little 5-year-old girl, her mother is a colleague of mine, and I was explaining in a lecture which the little girl attended what I've just explained to you, and she summarized it in this little drawing that she did for her mum later that night. Why do we dream? So we can have a good night's sleep. So that's my message. Whatever else dreams do, they appear to protect sleep, just as Freud suggested, using purely subjective psychological methods 120 years ago. So that's the story. I know there's a lot I've had to gloss over, and I'm looking forward to question time to be able to address any of the details that you would like me to go into further. Thanks very much.